Okay, good afternoon to everybody joining from the UK, and good morning to those of you joining from the Americas, and welcome to our final International Toxin Talk seminar, where we will be celebrating ECRs from the Americas. We've got 4 really great speakers coming up, 4 really great talks, so I will hand over to Anna Klompen to get us started.
All right, thank you. And thank you to everyone for being here at whatever time it may be. So we have 4 talks today, and just to get started off, our first will be Schyler Ellsworth. He is a PhD student in ecology and evolution at Florida State University, and the talk is titled Adding a New Dimension to Venomics, Evolution Mapped to the Molecular Structure and Scolopendra Morpha Venom.
All right, thank you. So yeah, like was said, I'm going to be presenting on kind of my first part of my PhD dissertation, which is looking at the molecular evolution in Scolopendra morpha venom. And so as introduced, I'm a PhD student at Florida State working in Darren Rakita's lab, where we work on a lot of different venomous systems. But today I'm going to be talking mainly about Scolopendra morphs, or our centipedes. And so to kind of get started talking about some of the molecular evolution that we look at, we're mainly looking for kind of these traces of positive selection. So just as a refresher, positive selection kind of fixes some of those beneficial mutations in populations and can promote the emergence of new phenotypes. So a great system to study this is venom, which we can see here with our eastern diamondback rattlesnake. And then up above, we can see the venom profile and all of these venom proteins and toxin families that we have in their venom. So venom is thought to mainly have evolved through this positive selection. So we have this positive selection that selects for these new phenotypes, and we can get the emergence of a lot of different types of venom proteins, even within populations or within new species as they emerge within venomous organisms. So for my talk today, I wanted to talk about some of the work that I've done where I'm testing for evidence of positive selection in centipedes across North America, specifically in the family Scolopendra morpha. So to start off, I'm going to be talking about some phylogenetics that I've done with this system. Then we'll get into the toxin diversity, and then more specifically talk about the molecular evolution in a specific protein family. So to kind of start off, I've been able to sample centipedes from across North America, where we've collected centipedes down in southern Florida on the Keys. So these island populations have the Scolopendra alternans. We've been able to collect a few local species from right here around Tallahassee, where Florida State is, where I've been able to collect Scolopendra viridis, Hemiscolopendra marginata, and Scolopogryptopsis spinosus. And then I've been able to collect a few different populations of Scolopendra heroes, which I have here in Texas and Arizona, as well as Scolopendra polymorpha, which I've seen in and collected in Arizona and also in California. And so we've collected these centipedes from across North America, and then we bring them into the lab where we're able to extract venom from them. And so how we extract venom is we have them kind of flipped upside down on this PVC pipe. And then we're able to have them bite onto and have their forcipules around a spatula. So the spatula then protects it from getting anything from their mouth, but we just get the venom from their forcipules. And then with a little bit of electrostimulation, we're able to get the venom to come out onto this metal spatula, which we can kind of see here from a different animal. And then we're able to collect this with micropipettes to use in later analyses. And then 4 days following these venom extractions, we're able to dissect out the venom glands and extract that for RNA, and then get the RNA-seq data that I have used for this talk. And so to start off kind of straight into the data from that RNA-seq data, I first wanted to be able to know which species were closely related, and that might relate back to how this venom has evolved within this group. So I have two outgroups that I planned on using. One of my outgroups is here on the bottom, Scolopacriptopsis spinosus. And then the other outgroup, which turned out to not be so much of an outgroup, is Hemiscolopendra marginata. So Hemiscolopendra marginata is a sister genus to Scolopendra. We can see here that with this molecular phylogeny that I generated from that RNA-seq data, that Hemiscolopendra marginata actually clustered within the genus Scolopendra. And so this kind of highlights some of the needed work that's some of the work that's needed on centipedes, that we're able to get some of this molecular evidence for their phylogenetics, so we can more accurately determine which species are closely related. So this tree was generated with about 1,000, I think 800 to 1,000 nuclear loci. And so this is a pretty good representation of what we currently have, but there's a lot more work that needs to be put into this area. So for the rest of my talk, I kind of separated these groups out into what I'll talk about later on with their venom. So I have the venom from Hemiscolopendra marginata that I'll talk about. And then Scolopendra viridis, we can see, is clustered pretty close with Scolopendra polymorpha. In Scolopendra polymorpha, with a name like polymorpha, we can expect that this kind of got split into 3 different lineages here. So I'm going to be talking about 3 different populations or lineages of polymorpha. And for their venom characterizations, these are the lineages that I split them into. And then Scolopendra heros, I also split into 2 different lineages. We kind of have the Texas lineage and the Arizona lineage that I split those into here. So like I said, there's a lot of work that still needs to be done in here, but this was kind of the first part of my project. So after this, I was able to look at both the proteomic data and the transcriptomic data to identify and annotate the different toxins in all of these species that I worked with. So I sequenced and did the proteomics for 26 different individuals. And here I'm showing the transcriptome toxin class abundances for each of those individuals. So I wanted to highlight a few of these different individuals where we have the percent composition for each of them here. And you can see that I've separated them out based on that phylogeny. So the first group I wanted to talk about was this group over here, Scolopacriptopsis spinosus. So we can see that this group has this yellow bar that I've labeled here as metalloproteinases that we don't really see in any other group. And so I was able to characterize this protein along with some help from my lab, where this protein is actually an adenomyosin-like metalloprotease that we haven't seen in centipedes before. So this is very common, is commonly associated with the snake venom metalloproteases and shares a very similar structure to those snake venom metalloproteases. However, this type of protease hasn't really been found in any other venomous species. And so this was kind of a cool result that we found from the venom composition of this one species. The other species that I kind of wanted to highlight that looks a little bit different from all of the other species is this one here, where we see some dramatic changes from the male and the female in the centipede species. And so again, working with some of the members of my lab, we were able to characterize this venom, but then also sample multiple individuals and look at their venom composition. And we were able to do a principal component analysis that I've shown here, where we have our male venom very distinctly separated from our female venom over here. So we do see a sex bias difference in some of the venom composition in centipedes. And so these are two of the different results that we've kind of already published from this data set. However, like I said, I've done the transcriptome and the proteome of all of these species. So I'm looking at doing a more comparative paper across the members of Scolopendra amorpha for North America. So the other part of my work that I've been looking at is looking at the molecular evolution of some of these toxin classes. And one toxin class that is very common in centipede venoms are these cap proteins, or this green band that we see here. And so with looking at this proteomic abundance, I decided to look at the molecular evolution of this cap protein. And so CAP proteins stand for cysteine-rich secretory proteins, antigen 5 and pathogenesis related 1 proteins, which is why we label them as CAPs instead of having to say this all the time. And these, like I said, have been recruited into a lot of different animal venoms, but principally in centipedes, they are pretty abundant. And they have diverse functional roles that have been talked about in a few different centipede lineages that we'll kind of highlight on this next page. So for analyzing all of the different CAP proteins for the members of Scolopendromorpha that I have data for, I generated a gene tree, which we can see here. So we can see that we have some relatively long branches here and some shorter branches here that are kind of more recently diverged. And so this closely resembles that we've seen in some of the other work that's been done in all of the centipede species, principally by Undheim from Australia when he was doing his PhD and postdocs there, where we have our CAP3 proteins, which we have this one representative and this one long branch on the top. We also have CAP1 proteins that are represented by this group here, which is mainly found in that out group. And then we have 2 different versions of CAP2 proteins. In CAP2 proteins, we have this group here that is typically trypsin inhibitors, is how they've mainly been classified. And then these CAP2-2 that I've labeled here are typically calcium channel inhibitors. And so for the selection analyses that I did on this protein family, I decided to focus just on these CAP2-2 proteins. And so I generated a different gene tree for this, and then I tested this gene tree for evidence of positive selection. And so I was able to find evidence of positive selection in this protein family, where I got evidence of positive selection with a p-value less than 0.001. So after this, I specifically wanted to test and see if there were specific branches in this tree that showed more evidence of positive selection than other branches. So through that analysis, I was able to identify this top branch here, that this branch specifically is undergoing some more positive selection than some of these other branches that we see listed here. So I wanted to look further into kind of the molecular evolution going on within this protein class. So I looked at specific sites across this protein to see where there was evidence of positive selection. So here on the y-axis, we have all of the amino acids for this protein. And then I have the negative log of the p-value for evidence of positive selection here on the side. And so here I found 5 different sites across about 1/3 to about half of the protein that have evidence for positive selection. So I wanted to look at this, these 5 different sites in a little bit more detail. And so how I did this was I looked at the 3-dimensional structure of this protein. So the 3-dimensional structure of this protein, I was able to determine using a program called Alphafold, which recently was released this last year, that can successfully predict the 3-dimensional structure and the foldings of these different proteins up to around 90%. So I was able to predict the structure of this protein, and then I was also able to map those 5 sites that were undergoing positive selection. As we can see here, these 5 sites kind of cluster all in the one area of the protein, meaning that this area of the protein is probably under a high amount of selection, where the rest of the protein might not be under that same constraint. And so, like I said, we still don't understand all of the functions of these different CAP proteins, especially in centipede venoms. However, this spot might be a spot that we can look at for future studies into how this protein functions. So these 5 sites I was able to map and look at. So kind of just giving an overview of the project, as I was able to kind of start looking at the phylogenetics of the species, able then to break that down and look into the molecular evolution that is occurring within this group. And then lastly, map some of the selection that's happened in one of these protein families onto the 3-dimensional structure. So some of the future work that I've wanted to do for this group and to finish off my dissertation is I want to look into the molecular evolution of some of these other protein families, as well as do some functional assays of this venom. And one of the functional assays that I do is injecting venom into fruit flies, where specifically I've looked at characterizing the evolution of venom resistance. And so looking at the coevolution of venom composition with venom resistance, and I'm using fruit flies for that work. So hopefully here in the next year or so, you'll hear more about me talking about the coevolution of venom and venom resistance. With that, I wanted to thank my lab. All of them are very integral and kind of helped me to form this project and help me along with this, especially Dr. Darren Rakita, who we can see here. But then I'd be happy to take any questions, or if you have any other comments, you can contact our lab or me at these links here in the bottom.
Great, thank you so much. Very cool stuff. So we do have a few questions in the chat, so several. So first, Charlotte asked, are you planning on conducting studies on the different isoforms of the cap protein related to their function? Do you have any hypothesis on what these different functions may be?
So I don't plan on specifically looking at the function of the different isoforms. So our work is mainly characterizing kind of these broad molecular studies, but this definitely is open to looking at some of the different isoforms. But I don't think for my dissertation, I'm going to be looking specifically at the function of the different isoforms.
All right, so just moving down. Did you find any sodium channel inhibitors in the venoms? I think they've been found in some Asian species.
So sodium and channel inhibitors, I don't believe I found a whole lot of sodium channel inhibitors. However, a lot of the scholop toxins have some diverse functions, and I haven't quite got to looking around to all of the different types of scholop toxins that I found in this species, in these species.
Are there any sites under selection overlapping onto where the caps bind to their receptors, or do we not have binding structures for those yet? From Dr. Dropik.
Yeah, like I was stating when I was looking at the three-dimensional structure, I'm not sure, specifically for centipede venoms, we're not sure of the binding structures, but that is something that I would be interested in looking at. And I'm not sure if those have been characterized in other species, but that's something I'm definitely going to look into.
All right, and then one more, and then I might have a quick follow-up to this one. So how did the cap family of proteins in the Scolopendra compare to those in other invertebrate venoms?
Yeah, so right now our lab has worked, like I said, with a variety of different species, and I haven't been able to compare the cap proteins from centipedes to, we also work with scorpions, spiders, and snakes. So I haven't quite been able to do that level of comparison yet, but I do have a few other projects that are coming up that we can look at that kind of evolution that's happening within that protein group.
Very cool. So my question selfishly is about a different family that I just noticed in your bar graphs there. So one of those was BPFT, which I assume is some sort of pore forming toxin. So those are something I'm really interested in mine. And I just noticed you had a lot of variability in those too. And I know you haven't done the functional stuff yet, but do you have any suspicion that those are having a pretty important effect in the venom function?
So those seem to be a very common family of proteins within centipede venom. So those are the beta pore forming toxins. Mm-hmm. And so there's some recent work saying that those potentially have entered the centipede venom through horizontal gene transfer. So potentially getting those from prokaryotes. However, there's not a whole lot of work that's been done specifically with those centipede venom proteins, but that again is another area that I'd be interested in, and definitely want to talk to you more about that.
Very cool. Well, great. Thank you.